Welcome to the July webinar from the Globe Mission Mosquito team. Please again use the chat if you haven't already to tell us where you're calling in from. I'd like to invite everybody to the Globe Observer Summer of Celebration 2022. At this free event, you will have conversation, you'll be able to meet other volunteers and contribute to the future of Globe Observer. There's a volunteer spotlight where you'll hear from a panel of Globe volunteers from India, the Philippines, Nigeria, and the United States. There'll be a science briefing where you can learn about the science accomplishments that are built on the data that you have submitted to Globe through the Globe Observer. A question and answer session, so if you have any questions for the Globe Observer team, those will be answered while you're there. And as a special registrant or attendee, you get an advanced look at upcoming developments and activities. Highlights from the volunteer observations will be a tour through the world, and you'll see what others have shared with their observations. And you may even spot one of your own. So please join us on July 26th at 7 p.m. Eastern Standard Time. The registration information is on the right-hand side of the screen. It's tinyurl.com/observer-c3. If you have any questions or comments, please be sure to put them in the chat. A quick update on the observations that you all have been contributing to this very rich database, but we now have accumulated 37,056 observations and eliminated over 22,000 habitats. So we're very proud of all of our observers, and the map kind of gives you an idea about where everybody's making observations from. Rusty, Andrew, and I from the IGES team of NESIC have an opportunity to be mentors for the Globe Mission Mosquito, the SEAS, the Earth System Explorer NASA internship. And this is, I think it's our 4th year, Rusty. And every year, these students that we work with are extremely talented, very top tier kids who are very inquisitive about science and STEM programs. This year, there are 106 that we're working with. And of those 106, we have made over 1,500 land cover observations and 451 mosquito observations. And why that's important is because we want to share today with you some of the projects that they have come up with as part of their introduction to Earth System Explorers. However, we're very proud of all of our Globe observers, and I'm going to let Dr. Lowe talk about this for just a moment, but we're like super excited and very happy with this. So take it away, Dr. Lowe.
Thank you, Cassie. So one of the things that participating in this citizen science project makes possible is the use of the data in research. And I'm really pleased to announce that yesterday, the paper that we had submitted to insects for their mosquito surveillance issue for citizen science was just published. And so we have a team of scientists that have worked on this article, including 2 C students from 2020. And we also have a mosquito abatement district person who's also one of our co-authors. But once again, these things are not possible. These scientific advances are not possible without the tremendous contributions of our citizen scientists, and particularly those people here in the Globe Mission Mosquito campaign that are working with groups and with classrooms and with teachers to get people all the information they need to contribute effectively to this program. So this is a shout out to you. Thank you so much. And you can download this paper at this URL if you'd like to read it.
Thank you very much, Rusty. I'm glad everyone's gathered today to join us on our webinar because I know that there's a lot of you that are working with students and wondering how you can get started in in an IVSS project, for example. And so our Cs, or system explorers, have some really great ideas and projects that they've created in the last 5 or 6 weeks with mosquito traps. And I just want to kind of give you the baseline of the whole project so that you can see where we're coming from. And I hope you have some questions because a lot of them are online and available to answer them for you. So we gave everybody the same basic idea, and this is their instructions and the kind of data that they're collecting. And so they were to build a trap with the following materials, and it was only a suggestion, but some container to hold water like a bucket. It could have been a soda bottle, it could have been a 5-gallon bucket, it could have been an old tire, it could have been lots of things. Suggested they put rocks in the bottom, use a piece of wood that would come out of the water for the female mosquito to lay her eggs on. They could pick a bait source, the type of water, and then since it's an experimental design that they would possibly be putting out in different locations, a sign identifying exactly what they were doing. And you can kind of see the directions to build the trap and the type of data that they were collecting.
And Cassie, we are going to also make this this protocol available on our website soon for general use.
Yes, I have it ready, Rusty.
I know you do.
So today I have the following students who are either online or have submitted a real brief video explaining what their take on the project was, some of the highlights that they gathered from it, and possibly next steps depending on how their project has been going. And one of our students, AJ, is online and he's created a story map that he'll explain in explaining his project, but a link to his story map is across from his name. So you're welcome to take a look at that too. But I'm ready to try the videos then. They should play one right after another, and then we're going to pause and ask for questions and comments.
Hello, my name is Paxton Calder, and I'm a C's 2022 peer mentor. I was an intern last year in 2021, and I used the Globe Observer app and Mosquito Habitat Mapper to conduct my own research. Specifically, I studied the effect of natural bait type on mosquito oviposition in central Virginia. My main research objective was to determine what types of vegetation may be more problematic in exacerbating mosquito-borne issues in my area, such as communal disease spread. I set up 3 ovitraps, each with a different bait type of the same quantity: pine needles, white clover, and tall fescue grass. I filled the traps with 3 liters of water, added a large rock for stability, and placed a smooth wooden ruler inside to accommodate species who lay eggs on such surfaces as opposed to the water itself. The traps were placed in a well-shaded and undisturbed backyard, and from the 10 to 12 day mark, depending on maturation, larval counts were totaled and the data was uploaded to the Mosquito Habitat Mapper function of the Globe app. Here's a view of the study site. As you can see, it's very green and lush, as well as shaded and protected, likely an ideal site for female pregnant mosquitoes to lay their eggs. As for interesting findings, I came across 2 Toxorhynchites elephant mosquitoes in the pine trap, which are cannibals that eat thousands of other larvae, meaning larval counts may have been much higher in the pine trap if the cannibals had not been present. But all in all, the pine needle bait did surprisingly well despite its acidity. This could be due to the presence of liminin and pinene in pine, to which mosquito antennae are sensitive and attracted. The white clover resulted in more larvae present on average, and it seemed to break down faster than the other baits, perhaps acting as a more readily available nutrient. However, there was a great deal of variation between trials, such as grass being the more prominent bait type in trial 1 and white clover being the more prominent bait type in trial 2, during which there was a large rain event. After precipitation, a chemical known as geosmin is activated, which we as humans recognize as the earthy smell of rain. Mosquitoes are very attracted to geosmin, perhaps explaining why there were increased larval counts for some of those bait types after that rain event. Lastly, pupae were only viewed in trial 3 when there was excessive heat. This demonstrates a small downside to fieldwork where weather conditions are beyond the scientist's control, but you can still find beauty and validity in your data. As for what I've learned, I now more formally recognize and appreciate citizen science as a meaningful way for people to contribute to the world of STEM from anywhere across the globe. I have learned about the implications of such work, such as how peaks of West Nile virus from CDC data in Virginia seem to coincide with years of great flooding from sources like tropical storms, potentially because of increased water source habitats for oviposition to occur. Through this experience, I have become more open and willing to learn about and get involved in new fields of study and to slow down and zoom in order to take a closer look at the world around us and to not let good science pass me by. Thank you.
Hi, my name is Ionis Ya, and I'm part of the NASA SEA's Globe Earth System Explorer Group. And here's what happened with my mosquito habitat experiment. So I can't say that my mosquito habitat experiment worked as perfectly as I thought it would, and there were a lot of caveats that I went through and a lot of failures. So this is kind of how it went down. So my first initial research question was, how does the depth of a mosquito trap affect the number of mosquitoes that I could find there? And so basically I had these three containers, one that was a very shallow container, one that was more of a medium size, and one that was a large bucket. The only large bucket I found was kind of dirty, and it was kind of hard to get off the dirt. And in each of them, I put a stick, fish food, water, and stones to weigh down. So after 1 week, my results were nothing. So in each of the containers, I found a couple of other dead bugs, and it was super hot that week. So the shallow bucket basically had no water left. And the large bucket was, because it was so dirty, it was kind of hard to see. So after another week of basically nothing, I tried to shift the focus of my experiment, and I decided to have a new research question, which was, how does the type of bait affect the abundance of mosquitoes? However, during this experiment, because the only size of container I had in many different forms was the small cup, there weren't that many mosquitoes that came, even though I tried many types of bait. There's sugar, there's a pork bone, some grass, and a leaf. So after this, after another quote unquote failure, I decided to kind of do a last try, and it ended up working. So this last try was basically just another basically a medium container that I just put on the grass near a couple of shrubs, and inside I just had grass and leaves, and I realized that it did in fact work. I was able to find many mosquito pupa, and they were all swimming around. There were about 30 to 40 of them, and I took a couple of close-up pictures with our microscope. And so the conclusions I found that even though my experiment wasn't very a traditional experiment where I went through a lot of different tries, I learned a couple of things. For example, the water should be clean. The reason why the larger container didn't really work as well was because it was so dirty. It seems that grass and shrubs are the better location. In my first location, it was under a tree on top of a bunch of rocks. And also medium and larger containers work better, while smaller cups won't work. Thank you so much for listening, and I hope you enjoyed it.
Hi everyone, my name is Isla Sumer. I'm in the 12th grade at the Hockaday School in Dallas, Texas, and I'm super excited to be sharing the results and how I've developed my mosquito trapping experiment over the past couple of weeks through the SEA's internship program. So first, a little bit about my experimental design and setup. So the overall purpose of my experiment was to determine how the quality of water in a particular mosquito trap affects mosquito laying and breeding. So I made 5 mosquito traps total in order to measure this, and they were all placed within a shady spot in my area of interest. So I had 5 traps with 3 different types of water. I tested tap water, filtered water, and pond water. And some of my traps were in one location within my area of interest, and some of them were in a different location. And I made my mosquito traps out of 2-gallon plastic buckets and used fermented grass clippings as my bait. So here's some pictures of my experimental setup. On the left, you can see the general setup that I used with the bucket, the paint stick, the fermented grass clippings, and some rocks to keep the entire setup stable. You can see the 3 traps placed at one location, and you can see the 2 traps placed in another location. So I wanted to test 2 different locations within my area of interest, and I just set them up like this, varying the types of water used in all my traps. So then on my results, so I've checked my traps every Monday since I set them out, and I've collected about 3 weeks of data so far. So each week I take 3 samples from my traps, count the number of larvae and pupa, and average that number. Then I take pictures of my larvae using a microscope and identify them based on their siphon. I record my observations in my notebook and in the Globe Observer app. So in the future, looking at my next steps, I plan to continue my experiment, but also test out a variety of different bait sources beyond just fermented grass clippings, which is what I'm using right now. So considering my results, overall, I haven't seen a clear difference in mosquito laying and breeding between all the different types of water I've tested, but I've learned a lot about identifying mosquito larvae, collecting accurate measurements, and experimental design, even though I haven't seen like a conclusive result that shows that mosquito prefer a particular water source over others. And here's some really cool pictures I've captured over the past couple weeks. So you can see the larva I captured on week 1 that has a short siphon. You can see a larva that I took a picture on week 2, where this was my first picture. I was able to get one of the whole body of a larva, and so I found it super cool. My favorite picture I've taken so far is this third one because I really like how it has that long siphon. And I've also had the opportunity to practice identifying these mosquitoes based on their siphon types. And there's a little bit of learning curve, but I felt like I feel much more comfortable now. And then finally, a mosquito pupa that I took a picture of on week 2. So this was the first time I was able to identify that. And I feel like I learned so much about mosquito behavior and breeding and larvae especially. And this has been a really cool experience so far. Thank you.
Hello, my name is AJ Caesar. I'm a rising senior at Ridge High School in Basking Ridge, New Jersey, and my mosquito trapping experiment is titled, What Trap Should I Use? Determining what style of mosquito trap is most effective for reducing mosquito populations in relation to the cost of creating it. And pictured here on the right, I have the 4 different traps that I experimented with. So when I was first introduced to the Ovianta trap at the beginning of this internship by Dr. Lowe, I became immediately set on building one myself. But after 2.5 hours in my grandfather's workshop and numerous cuts all over my hands, I realized just the difficulty and time that goes into producing an Ovianta trap. On my drive home, I couldn't help but feeling like I would have been better off just making one of the bottle traps I had seen online, which sparked the inspiration for this experiment, which aims to answer the research question, how will 4 different traps with varying levels of input, time, money, etc. required to make them, compare in terms of how many mosquito larvae they're able to capture. And the idea is that if I were a civilian aiming to control mosquitoes in my backyard, I would want to use the trap that produces the most bang for my buck, catching the most mosquito larvae with the least input required. And all 4 traps were placed in close proximity, so location would not alter data. And here are my 4-week results. The first trap did not catch any mosquito larvae, but the other 3 were all able to catch sufficient quantities. Here's a photo of an Aedes mosquito larva, which you can tell from the short and blunt siphon. And here is a photo of a pupae of the same species. So some qualitative observations I made were that despite putting rocks in the bottom of the first trap, it just did not have enough size to withstand the elements. Every day when I went to check it, it was knocked over. And I concluded that this 16-ounce water bottle is not effective for making a trap. But one interesting thing I did find is that if you look at my data, week 4 for the Ovianta trap was the only instance where the quantity of larvae decreased throughout my entire experiment. And this was because another organism had taken over the trap, which I have videoed here. It is a worm-like organism with similar size and structure to a larva, but it behaves very differently. And this leads me to believe that it may have outcompeted the mosquito larva. Moving forward, I'm looking for ways to research, discover the identity of this organism, and eventually combat it. So what are some conclusions I've made through these 4 weeks? If I were advising a civilian on what trap to use, I would suggest that investments that are definitely worth the time and cost would be 1, hanging the trap using a string or rope, as this prevents ants from invading it, and 2, using a container with at least 32 ounces of water, because this proves most effective for catching mosquito larvae. But the investment that sparked the inspiration for this experiment, whether the timely and costly investment of creating an Ovianta trap is worthwhile, is still inconclusive due to the influx of planaria worms into the week 4 Ovianta sample. But moving forward, I'm confident that the durability of the Ovianta trap will prove its worthwhileness. Thank you for listening.
Hi, I'm Marvel Hannah, and I'm a NASA Seas intern in the Globe Earth System Explorers team. So my research had 2 purposes. The first purpose was to see if Ovi traps or safe bottle traps attract more mosquitoes. And the second purpose was to see if CO2 traps or CO2 traps under light at night attract more mosquitoes. So I did this research because studies have shown that mosquitoes are actually attracted to carbon dioxide and go to places with more carbon dioxide. And pregnant women are actually a feast for mosquitoes because they have higher body temperature during their pregnancy. Additionally, without sunlight, mosquitoes are attracted to light with high amounts of UV, but during the day, they like to stay in shady places to avoid the sun. And if these traps are successful, they're low cost, easy to assemble and maintain, and they're pesticide free. So these are my traps. The one on the top are the save bottle traps. This is a control, and this one is the light with the yeast, and this one is just yeast. And the bottom ones are the OV traps, which was also the control, light and yeast, and yeast. So in total, I had 8 traps. I had 1 save bottle trap and 1 OV trap with yeast. I had 1 save bottle trap, 1 OV trap with yeast and light at night. And then I had 2 negative controls, which was an OV trap and a save bottle trap with tap water. And the negative controls were just to make sure that they didn't have any mosquitoes or mosquito larvae and pupae. And I also had 2 traps in my area of interest that were save bottle traps with pond water and grass clippings. These are the traps in my area of interest, and they were inside bushes. And the results. So I found fruit flies, and even in the trap of my area of interest, I had a spider, but I've not yet found any pupas or larvae, but there's still weeks to come, and I'm excited to see what happens. Thank you.
Hello, my name is Emily Wang, and I am one of this year's NASA SEAS Earth System Explorers interns. So my research aimed to answer these 2 questions. The first question is, what is the effect of color on the number of mosquito larvae found inside various traps? The second question is, what is the effect of sunlight on the number of mosquito larvae found inside various traps? So for my experiment, I decided to set up 6 traps. I painted 2 traps red and 2 traps blue. Then I placed a white, red, and blue trap directly in the sunlight. I placed the remaining 3 traps in the shade. I filled each bucket with 3 liters of water and 100 grams of weeds. A wooden stick was also placed in each bucket and used to stir the contents of the trap together. When I checked my traps the following 2 weeks, I noticed a significant number of mosquito larvae in several of the buckets. Although I was not able to get a precise photo, I believe the larvae to be of the Culex genus. My initial finding after these 2 weeks saw that there was generally a larger number of mosquito larvae in the traps located in the shade. Additionally, traps that were in white or red buckets contained more mosquito larvae than those that were in blue buckets. However, as I observed my traps, I noticed that there was a pretty big issue. The paint that I had initially applied to my buckets had started peeling off at the bottom of several of my traps. This presented a major issue since the color variable of the experiment was no longer consistent across both groups. Additionally, I could not be sure of what chemicals were in the paint that could be contaminating the water. Thus, I decided that it would be best to create new traps that were already different colors. I wanted to avoid painting since it had already shown to peel off. I was unable to find containers in the exact size and colors of my previous traps, but I decided that it would be best to pivot the experiment in this new direction given the problems that I previously faced. Today, 1 week after I set these new traps, I checked them and did not find any mosquito larvae. However, I'm hoping that with these new traps, I will be able to gather more precise and accurate data in the coming weeks. These setbacks definitely showed me that problem solving is a big part of science and experimentation. Through this process, I learned how to keep an open mind and just be willing to adapt to my experiment. I'm excited to continue my research in the following weeks and monitor my results. Thank you for watching, and a special thank you to all of the SEAS mentors for their guidance throughout this process.
My name is Joanna Pledgy, and I am one of the SEAS Earth System Explorers interns. My mosquito habitat experiment is focused on answering 2 questions. The first question being, how does container width and depth affect mosquito oviposition? And the second question being, how does container bait type affect mosquito oviposition? Specifically, do mosquitoes prefer naturally produced or artificially produced nutrients? I'm answering these questions to understand which container and bait types promote the most mosquito oviposition in my region, to potentially apply this research and data to public health. My experimental setup consists of 3 types of traps, with 2 traps per each type. To enunciate, I placed 2 safe bottle traps, 2 bucket traps, and 2 pie tin traps, for a total of 6 mosquito traps. For each type of trap, 1 trap was baited with grass clippings, and the other trap was baited with cat food. The grass clippings are acting as my naturally produced source of nutrients, and the cat food is acting as my artificially produced source of nutrients. For instance, one of my safe bottle traps was baited with cat food, and the other safe bottle trap was baited with grass clippings. I also took careful consideration in keeping all other variables controlled, such as the general placement of each trap, the trap size to water ratio, the trap size to bait ratio, and the colors of each trap type and their components. My traps were first placed on June 13th, and since then I've collected very intriguing data. I check my traps weekly, and for the first month, only my large bucket trap baited with grass clippings produced any mosquito eggs or pupae. Each week, I was able to ID these pupae as those of the Culex breed. Also during this time, all of my traps baited with cat food were only found with dead bugs, fly larvae, and even slugs in them. This data led me to believe that the cat food may contain a preservative that was killing all of these insects and thus deterring mosquitoes. However, this past week, I experienced a potential research breakthrough. After being unable to check my mosquito traps for nearly 2 weeks due to vacationing, I returned home to not 1, not 2, but 4 of my 5 still standing mosquito traps hosting Culex eggs, pupae, and adult mosquitoes. 2 of these traps were baited with cat food, which surprised me beyond end and has led me to ask new data-based questions that I hope to be able to answer as I complete my mosquito habitat research in the coming weeks. Thank you so very much for taking the time to view my recent research progress. And if you have any questions at all, feel free to email me at the contact provided below.
We have one live presentation, so I will stop sharing. And Aiden, I think you'll be up next then.
Yeah, hi. Let me share my screen and we'll get started. All right. Hi, everybody. I'm Aiden Schneider, and I'm a rising junior at the Davidson Academy of Nevada. So I'm located in Reno, Nevada, and the climate here is semi-arid. Nevada is actually the driest state in the United States. And Reno's daily temperatures vary significantly, with June having a high average temperature of 29 degrees Celsius and a low average temperature of 10 degrees, and July having 34 and 13. Mosquitoes tend to breed best at around 29 degrees, so there's a large variation of temperature between from this best breeding temperature. So it's also a very sunny climate with low vegetation due to the semi-arid climate, and Reno has very shady areas as a result. There's also 300 days of sunshine a year on average. Mosquitoes are not commonly found in Reno, and a prior SEAS intern located here in Reno supported this assumption by logging no mosquito larvae throughout the internship. She only found a few adult mosquitoes on the final day of logging in August. So my methodology was I have containers of 5-gallon Home Depot buckets colored orange. Each have a wooden paint stir resting inside the bucket. About 1/4 of each paint stir sticks out of the water so as to provide a location for mosquito oviposition. Containers are lined up one after another and are all located in a shady area on a 3rd floor balcony. I placed them on a 3rd floor balcony because that ensures that the traps are in a shady area and are undisturbed, so then the still water environment can be maintained and are not subject to any pest control programs. In my experiment, I'm investigating whether various types of bait make any difference to the baseline of very few to no mosquitoes expected in Reno. I have 5 different mosquito habitats, each with their own bait. The mosquito habitats contain tap water, and this is my control variable, pond water collected from a nearby park to emulate that nearby habitat in my residential area, hamster food mixed with water, fish food mixed with water, and grass collected from the same park mixed with water. Now, my hypothesis for this experiment is that since mosquitoes are commonly found in nutrient-rich water with lots of food for larvae and pupae, the mosquito habitat with pond water will have the highest total number of mosquito larvae by the end of this experiment. I'm checking these traps every 5 days to see if there are any developments in mosquito count. My mosquito traps are located in the same area as a previous C's interns mosquito traps. By using a comparable methodology in my mosquito traps, I aim to provide continuity in the Globe MHM dataset for this area as to improve its usefulness for research using citizen scientist data. Thank you for listening to my presentation.
Thank you, Aiden. I'd like to pause now and ask if there's any questions from anybody.
I think it would also be great for the C's interns. If you have questions for the other interns, that's also appropriate here too. I was just really, it's just so interesting. There are quite a few blogs that have been written, and it's been interesting for me just to look at all the different variables that have been examined this year. I think this is kind of a record, Cassie, isn't it, in terms of the range of
Right.
variables that have been explored. And this is just this is a very nice representative handful of them. But if anyone has any questions, please go ahead and put it in the chat or raise your hand. We'd love to hear from you.
I do have the blogs, Rusty, for you. If you wanted to run through those, I can share.
Let's do that real quick.
Okay.
I think one or two of them, well, one of them at least is the same. We've already presented live, but what I did was I wanted to share with this audience the fact that we do have these blogs. Right now, they're on a private space because what we've been doing with the interns is we gave them a blog area, and then as they go through their project over the 3, 4, 5 weeks of their experiment, we asked them just to update these blogs. And so here's one by Michael, and he's looking at the effective container surface area on mosquito oviposition. And he is coming up with his outcomes, but you can see here below that that we have another wonderful example of an egg raft, as well as a very nice mosquito specimen in these containers. So that's one thing that can be done, is looking at the same color container, but different depths of water. Thank you. And different surface area. Okay, next one, Cassie. Another one that I pulled out was one by Ishan, who is looking at water quality and satellite imagery. And so what they are doing is they are looking at the effect of water quality on the habitats that are found of mosquito larvae in water bodies. And so he's been doing an experiment just walking around to all the natural water bodies and making observations. So it doesn't need to be a trap experiment. You can also just do a local walk and make some observations. And that is also a perfectly appropriate research project. Next one, please. One of the reasons that his team is doing this observations of these natural surfaces is one of the things they're doing is they're going to be using some of the they're using a little bit of they're developing a machine learning model where they want to predict cases of West Nile virus and see if there is a relationship of this disease to the greenness or the color of the water bodies, with the color of the water bodies being a proxy for the presence of algae. And so they just a couple of days ago, they just published some of their preliminary data on the blog site, where they are seeing that they have been able to distinguish water coverage using their methodology. And we are staying tuned to see what they will be able to do for this. Next one, please. This one is by Raymond. And what he was interested in doing, it was looking at how sunlight and container size affects mosquito oviposition in terms of how many larvae and what kinds of larvae are found in each container. And what he's found so far is that the mosquitoes definitely are more attracted to the shadier areas and the larger bodies of water. And then he even posits that maybe an underlying variable for this might be temperature, because larger and shady water bodies tend to be cooler. So we don't know if that's the case or not, but that's a really interesting hypothesis to explore. And he also noted that he sees, he's looked at the mosquitoes there, and he's identified them to Culex pipiens using local taxonomic keys. Okay. All right. Here is one, a very interesting presentation by Sarah Beckett. She's testing the effect of fertilizer on mosquito breeding. And she lives in Tampa Bay. And as part of the background for her experiment is that, there is a lot of runoff from agricultural areas as well as lawns when it rains. And when this happens, the runoff that contains these nutrients go into the water bodies, and they begin to impact the life that are in the water, namely algae blooms. And these algae blooms are very, can initially be helpful, but ultimately might be harmful if you get so much, overgrowth, for instance, of algae in a water body. Then what happens is there's too much algae, so you get too many fish, and then when you get too many fish, during the night when there's no oxygen production, the fish actually starve from oxygen, so they basically suffocate, and then they die, and then you get like a dead water body. So that is part of that eutrophication process that takes place. Other times you might get the development of toxic algae, like we have, like for instance, she mentions here, red tide. So anyway, she's very interested in this question. And in her research, she has set up 4 buckets, and she added fertilizer to each one of them. And after a week, she says she found 250 mosquito larvae in a bucket with 2 teaspoons of fertilizer. Yay, science. So this is an ongoing project, but I just love that yay, science. Next one, please. Okay, thank you, Cassie. So the reason I wanted to show these blogs is right now these are on a private space, but once the blogs are ready for public consumption, we will be posting them on the Globe Mission Mosquito blog site, and you'll have access to maybe 30 or 40 experiments that you can look at and utilize as a way to sort of generate ideas for research that you might want to do in your community, on your own, with students, in camps, and so on. So thank you so much for listening to these talks today. And I also want to, from the deepness of my heart, I want to thank these incredible SEAS high school interns, these research interns that have been doing marvelous work throughout the summer. So thank you for contributing your time.
I also would like to thank the interns for taking time out of the day for both making their video presentation, recording their data in such a great, understandable way, and then spending the time with us on our webinar today. If there are any comments or questions from the attendees for anybody, please put them in the chat. We'd love to hear what you're thinking and how we can go further with their research. Thank you, Inez. We appreciate your positive comment. August 11th will be our next webinar. And again, if there are any comments or questions, you can always review our webinar, and you can contact me and I'll get in touch with whoever needs to answer your question. And thank you. So maybe the SEAS kids would like to turn on their cameras and we can say thank you very much before we sign off.
Thanks, everyone. This has been really, really wonderful. I do appreciate so much you giving of your time to share your information with our international citizen science research community. And just as a note to everybody who is here today, if you have any ideas for things you'd like us to do in this webinar, right now, Cassie and Liz and I are beginning to come up with topics for the future webinars for this upcoming year. We do have our international advisory board, which gives us ideas, but we are also interested in your ideas. So drop us a line and let us know how we can support you in doing your own citizen science research. So we hope to hear from you.
All right. Again, thank you, and we'll see you in August.